Thank you, Tony, for that very nice introduction. Thank you for inviting me. It's a great honor to be here and also to be among so many of my friends. Last year I gave a talk on immunology, so I thought this year we would talk about other things, and we're going to talk about virulence. But first I thought we should say something, I should say something about Dr. Kinian. This was a beautiful article written by Dr. Morris and Fauci last year, which is an M-Bio, you can read it. And this was a remarkable individual whose life saw the germ theory revolution. And so he went from being a physician who couldn't do anything about infectious disease to be able to treat it with serum therapy at the turn of the century. He saw his daughter died of diphtheria, and yet during his life saw a treatment for diphtheria, perfected a strain for tuberculosis, and combated the plague outbreaks in San Francisco through public health measures. But when I look at his life, what I see is that it spawned a revolution. It spawned one of the great human revolutions in that physicians were able to associate diseases with specific microbes. And it's not unlike the revolution that some of us are living in, where we went from computers being things that were far away to being in every part of our lives. I'm going to begin with some existential questions that are not part of any usual seminar. Why are we here? Why are we so hot? Why do we eat so much? And why, when you drive in the Maryland countryside, most of the large animals are mammals? Now, you would argue that these are important questions, yet you see them addressed in seminars. Now, I'm going to give you the answer so that you don't have to stress out. It's a single word, and it's fungi. And I hope to convince you at the end that this all makes sense. So I have some other goals. One is to show that science can be fun, and the second, to argue for much thought in biology. In biology, we don't think anymore. In fact, we can't think without data, and I think that's one of the things that we have lost. So infectious disease before the germ theory. The explanations, miasmas, contagion, public law, punishment. Diseases will come and go, and people would die, and there was very little anyone could do. The germ theory revolution begins with a technological innovation, the microscope, but it takes 200 years between microbes that are associated with diseases. Causation established in the late 19th century, and then within 6 or 7 generations, many of the diseases that kill humans since we have had history tend to go away. So we have intervention, sanitation, serum therapy, vaccine, antibiotics, and then mechanism, and people have been working on mechanism pretty much ever since. So an early problem in microbial pathogenesis, are pathogenic microbes different? Well, in the early days, it appeared to be so. They made toxins and they had capsules. But there was a divergent view posed by Bourdet at the Pasteur. He is the one who discovered Bordetella, and he also contributed tremendously to complement, that argued that there was no fundamental difference between pathogens and non-pathogens. Virulence was not a stable trait. You could take an organism, attenuate it, and bring it back to virulence. And also, immunization negates virulence. Then it became a very confusing evolution in thought. From the 1670s to 1860s, the microbes were seen as small animals. Then we separated them into pathogen and non-pathogens. And by the late 20th century, we had tremendous confusion. Things were called pathogen, opportunists, commensals, and non-pathogens. And I would argue that ultimately this last business is largely an error in thought and error in logic. So the problem begins as these things can move in one direction or the other. Pathogens could become non-pathogens, as alluded to by vaccination. By the 1990s, it depends who you ask. If you ask a microbiologist, how do you make a pathogen? They say, take a microbe and put something on it, like a virulence factor. Immunologists will take a microbe and make a susceptible host, but neither had the story. For immunization negated virulence. Variola major is a non-pathogen in hosts given vaccinia. And on the other part of the coin, for example, patients with AIDS do not get all microbes. So the problem was that neither view could account for it. But in fact, the central problem was that a microbe that is pathogenic, when you call a microbe a pathogen, you're giving it a trait that is not its own. For a microbe is pathogenic only in a susceptible host. So we set out to try to reconcile some of this in the late 90s, and in particular some of the papers that appeared. And you can put all microbial pathogenesis, all symbiosis, commensalism in this diagram. And if you focus on the arrows where the host goes down, that is where microbial pathogenesis will be at. I worked with Lisanne Porofsky, and we came up with the damage response framework. This was originally a teaching tool. We used it to teach graduate students. And after a while, we realized that in the teaching notes, we had original thought, so we went on and we published it. So it's very simple. You have two entities, a host and a microbe. You have, in this case, the relevant outcome is damage, and damage can come from the host, the microbe, or both. And the insight was that there had to be a relationship. And after doing a lot of reading, we realized that the relationship is a parabola. People get into trouble at either end, and if you drag it below the x-axis, you could see an organism that could be beneficial at a certain immune response can become pathogenic in a setting of a very weak immune response, or alternatively, if you have a disproportionately strong immune response. So how could you use it? Well, imagine Saccharomyces cerevisiae. You can buy this in the store. You don't need a license. You don't have to file out any applications. Yet, if you go and you look in PubMed, you may find that this causes disease. So it causes disease in patients with AIDS. In the old days, now we don't see this anymore. So you're going to call it opportunistic, but you keep reading, and you find that it causes identical disease to Candida vaginitis, and this is in normal women. So what are you going to call it now? A primary pathogen? And here is a baker that has long taken care, a long taken out because of massive inflammation, a problem at the other end. So if you look at the damage response, you can put the AIDS patient at one end, the baker with the nodule at the other, and I don't know where normal women with vaginitis would be, whether they would have a toll receptor polymorphism or an abnormally strong response. The damage framework also intellectually allows you to transcend a lot of these things that are confusing. For example, if you look at damage as a function of time, we see all these states are continuous except for the amount of damage to the host. So commensalism to us is different from disease only in the degree of damage. And you could see, for example, if you were to plot Candida albicans in this, that basically we get infected, we become colonized, we may get it as neonatal candidiasis, then it gets controlled, but it's in life, it's basically moving along the states. The damage framework has some important consequences if you accept it. It shifted the base from pathogens to non-pathogens to the outcome of the interaction. According to the damage framework, there are no pathogens, there are no commensals, there are no opportunists. I see I misspelled it. It is amazing how you can go over talk over and over again in the computer and never catch the errors. And then when it's projected, they are apparent. And I think that's fascinating. And so virulence is one of the outcomes of the interactions. To us, there are only microbes and hosts. The outcome of states are continuous, and they differ only in the amount of damage incurred by the host. And we see virulent as an emergent property, and I will come back to that. But it basically frees you to begin to think very differently if you think only in terms of microbes and hosts. So one of the things that it does is that one of the problems in microbiology today is that we're living in a microbiological archipelago. All these fields are out there. They barely interact with one another. In fact, most of microbiology becomes no different than stamp collection. If you look at Darwin, Darwin looked at patterns. One of the things they tell you today is to focus. If he had focused on the pigeons, or on the shells, or on the finches, or on the insects, he would have missed the theory of evolution. What he did is he focused on the patterns. This was all thought. He had done very few experiments. It's all descriptive. There is no mechanism, and it will not be publishable today because he had no mechanism. And that actually tells you something about the state of science today. Now, if you look at the end of the 19th century, you would have seen a lot of these diseases. Some of them have gone away. Some of them are very much with us. But there would have been one big difference from life today, and that is invasive fungal diseases would have been extremely rare. You dial forward 100 years, and you see these diseases are very common. The question is, what happened? What happened is that we changed the host. So we had the medical progress, gave us antibiotics, altered the flora, IV lines created portals directly into the circulation, immunosuppressants, chemotherapies, ICUs, and then we had the cataclysm of AIDS, such that until recently, Cryptococcus is one of the leading causes of death in Africa, eclipsing tuberculosis, and yet you don't hear about it. 2 days ago, we had a New York Times editorial, and it cited the big killers. Fungal infections, certainly not there. Fungal facts. These are the most successful kingdom with perhaps over 1.5 million species. They are the major pathogens of plants, reptiles, invertebrates, and amphibians. Very few species are pathogenic for mammals, and invasive fungal diseases in mammals are actually rare, and those with intact immunity. However, fungi are devastating entire ecosystems, from corals to the frogs. The amphibians, incidentally, made it through all the major extinctions of life, and they are now in trouble from a single fungus. Rice, salamanders, turtles, wheat, the list can go on and on. So what the pattern there is, all these hosts are ectothermic. Here is a host that is in trouble from a fungal infection that arrived in the United States in 2006, white bat syndrome, but it has no problem in the summer. In the summer, the organism is warm, but in the winter, the temperature drops to 10 to 12 degrees, and they become susceptible to this organism. We're going to see extinctions of several bat species as a result of this organism, and there is not very much that anyone has managed to do about it. So why are mammals so resistant to fungal diseases? And could the answer be in the distant past? Well, if you look at the distant past, what is very apparent is that the KT event, that is the event associated with the demise of the dinosaurs, so the replacement of the reptilian megafauna with a mammalian megafauna. Why, when you drive in Maryland, you see primarily large mammals and birds. And you can see that there was a reversal between ectotherms and endotherms, such that the organisms that have made it through this event were largely warm-blooded. So there are three questions that have troubled me, and I ask, could they be related? If the reptiles were so fit, why did they not reclaim the Earth? They need about 1/10 the energy that you do. If you were reptilian, you would probably have to eat once a week. They have many progeny. They don't care about their progeny, and they do extremely well. And why are there so few pathogens for mammals? And this all related to why is medical mycology so undervalued relative to virology, bacteriology, and parasitology. So I'll leave it at that. But you can see how my own issues drove some of this. So adaptive immunity cannot explain the paucity of fungal diseases. All vertebrates have adaptive immunity with cellular and humoral arms. The reptiles and the amphibians have adaptive immunity. The reptiles and the amphibians are highly susceptible to fungal diseases, and I mentioned the catastrophic fungal declines. Now, work by John Perfect and others gives you a hint. If you take a bunny and you infect it with Cryptococcus, it doesn't matter where you put Cryptococcus in it, it clears infection. But if you put it in the testicle, one can get a chronic orchitis that does not disseminate. If you give steroids, one gets a dead bunny, and that begins to suggest an emerging pattern that perhaps mammals has adaptive immunity plus endothermy resistant to fungal diseases. So I began to work with this individual, Vincent Robert, and a few years ago he designed bioinformatic tools and looked at thermal tolerance of 4,000 fungi in one collection and came up with that relationship, such that you can see a very dramatic drop in viability as you go from 30 to the 40s, such that for every degree of temperature, one can exclude roughly 6% of fungal species. Then we took that data and worked with Aviv Bergman, who's a mathematician, and solved those two equations together. The benefit being protection against fungal diseases. The cost is the fact that the hotter you are, the more you need to eat. And look at the temperature that came out as the maxima. The optima appears to be a mammalian temperature. Now, it could be coincidence, but it is in 27 and it is in 48. It happens to be very close to your core temperature, suggesting that there may be more to this. So what killed the dinosaurs? Well, it depends who you ask. It used to be that people thought it was small mammals that ate the eggs. That absolutely makes no sense. I found this in the web, which actually makes no sense either, but it was from a criticism, the anti-smoking program. It could be increased volcanism. There was a lot of volcanism right about 100,000 years before the asteroid hit, and it could have been a two-hit a scenario where it was volcanic, and it could have been the dinosaurs. Now, the next one is true. This was, believe it or not, published in Current Biology last year. Somebody calculated that the large dinosaur mass could have triggered global warming through putting out the amount of methane to get a runaway climate change. So, and then there is one more explanation that I'll show to you, but I won't comment on it because I have no data for or against it. So The Cretaceous world. The Cretaceous world was a warm volcanic, it was a warm volcanic and forested planet. The Atlantic was very small. You had a reptilian megafauna, and it was a Yucatan impact, and this is what created the cenote ring. So next time you go to Cancun and you see the cenotes, they occur only there because basically the limestone and everything ended up getting melted. What was the post-impact world? Overnight, you had fires, smoke, and the dust obscured the sun. Photosynthesis was shut down for 6 months, and global temperatures dropped. There is pretty good data for this. There is also good data that there was a global fungal compost. So imagine a world, a forested world where all the trees come down. You would have a massive compost. You would have had massive spores. And I could imagine that organisms that were warmer may have been susceptible to the massive inocula that they would have been exposed to. And I wrote this a few years ago, tongue-in-cheek, but after more I thought about it, the more it made sense. So if you think about the post-impact world, and I hear, by the way, some of this beautiful art comes from Julie Marquardt, and she shared it with me when she was making the poster, so I benefited in multiple ways from coming here. So the reptilian megafauna was killed by the impact blast, disruption of food sources, the chilling of the planet, and maybe fungal diseases. Now you say to me, but aren't the dinosaurs supposed to be warm-blooded? And I said, sure, but they are warm-blooded because they are sacs of cytoplasm. As soon as you stop feeding them, there is no evidence that they could have regulated their temperatures the way you do. Reptiles usually fight off infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases. They can actually go right through the shell. And in fact, another group has picked up in this in China and shown that endolithic fungi attacking dinosaurs. Now, this may not obviously be from the KT event, but at least it's more circumstantial evidence. So some small reptiles survive. If they were so fit, why didn't we not have a second reptilian age? And I argue that there was a filter pretty much at the KT boundary that was a great global compost and selected for warmer organisms. And here is another one of Julie's designs, and I think she catches it very nicely. You have the reptilian megafauna, fungal selection create a filter for the emergence of mammals, and now you have the mammalian megafauna, and now these organisms were selected for it, and that's why they are so resistant to what is one of the major types of pathogens in the planet. So as to this hypothesis, I have no proof. Is it plausible? I think so. I have no experiments. It's all thought. It's a lot of fun. I think it's internally consistent. I think it's satisfying to me. I have no idea if they're accepted, and I frankly don't care. I never have to apply for funding on it, so it's something that and this is why I went into science. Science should be fun. We should be able to think and write about it and debate things, and this is something that you often don't see too much anymore. Incidentally, it's all written down in PLOS Pathogens in a few pages, that story. So now I'm going to focus on Cryptococcus uniformis, and I'm going to talk about that for a little while, tell you some of the work we do on it. And here I want to acknowledge the mycology intramural program. Jack Bennett, Jung Kwan Chung were enormously helpful to me when I began. And a lot of the work and a lot of what we know about this organism was in fact generated here. This is an organism that actually, as you can see, the pathogenesis is very similar to TB. Everyone is infected, but few get disease unless one becomes immunosuppressed. It's also a pathogen of plants, so it's a transkingdom type of pathogen. It has a very complicated life cycle that we have been working on for about 20 years, many papers from us and other groups. But what I want to show you is that it can pretty much, it can get into a macrophage and it can lyse it, and you're going to see that it bursted, it's dead, and part of the reason is the macrophage is not moving. This is a much more interesting movie. What you see here is a macrophage, and the Cryptococcus are replicating inside, and what it's going to do is they're going to come out, and they're going to leave the macrophage alive. And what I would ask you is to stop for a minute and think, why should a soil organism that has no need for mammalian virulence have a strategy like this? And if you don't believe me that cell's alive, keep watching. Going to replicate, and it's going to walk off, one of them walk off the field. This is sophistication, and yet this is learned in the soil. If you remove all animals from the world, I am sure this organism will still have that capacity. So Today we live in a sad time, because if you have a movie like that, you can enjoy it for about 5 minutes before somebody comes up to you and says, what's the mechanism? What's the mechanism? What's the mechanism? And if you answer that question, they say, does it happen in vivo? Does it happen in vivo? Does it happen in vivo? Well, this is reality. We have been working on the mechanism. It's complicated. It looks like we took a hint from virology. It looks like this may be a big autophagosome in which it allows fusion such that it doesn't lyse it. It does happen in vivo, and this led to a very complicated, that slide is 5 years of work. Basically, we had to learn how to label macrophages, load the macrophages, put them into a mouse, take them out, and you could see in the bottom about half of them were emptied when 24 hours later. So this mechanism is out there, and where does it learn to do it? The organism learns, we believe, to do this in amoeba. And if you look at the endolytic exocytosis can be demonstrated in amoeba. You can demonstrate pretty much everything that happens in a macrophage in amoeba. This, you can do it with dictyostelium too if you want a genetically tractable host. And if you look at evolution, these organisms were interacting with each other for at least 500,000 years before the first type of animal emerged. And so when the animals emerged, some of the fungi were there already having learned to be pathogens. If you look at expression, and I like to think that these two are separated by over 1 billion years of evolution. Look how the gene expression of the organism, once it goes into them, it's almost identical in the genes that it upregulates. There are more differences in the downregulated genes, but this type of conservation suggests that, in fact, are common origin. There are two major virulence factors, the capsule and melanin, or the lacase system, that have been studied here. Peter Williamson has done a tremendous amount of work on that. And they contribute about 40% of the total virulence. Incidentally, this kind of analysis has only been done for Cryptococcus neoformans. That is, there is different ways in which you can weigh the value of different virulence factors. And people may be working on a virulence factor that gives you all or none, but it may be contributing only 1% to the total virulence, because it may be needed, but what it tells you is that a lot of the virulence is coming from these two agents. The polysaccharide accumulates in vivo. Here is a patient that died. All the brown material is polysaccharide. This is what is measured in the serum as cryptococcal antigen. It gets into the macrophages and it messes them up. And this is a striking picture of a cryptococcal biofilm in an AIDS patient, and notice the complete absence of inflammation. And part of this is the difficulty of treating these diseases with fungal agents alone. There is no immune system to help you. Now, if you look at the capsule and you were to plot molecular mass as a function of ignorance, you find that we know a lot at the molecular level, and we know a lot of what we can see. The problem is in the middle. The problem is the capsule is one of those structures that is very hard to study. And I give you an example why. This is an India ink preparation, and look what happens when you dry it up. These are the structures are 99+% water. So you need to have different approaches. So I originally trained as a physical chemist, and I need to tell you a little bit of how we think. The physical laws are known. You don't go into a physical chemistry lab and question the second law of thermodynamics. You accept it and you leave it alone. The approach is to make accurate measurements. The measurements define the parameters of the system under the study. You use it to generate testable models, and then reality is approximated by models that resist falsification. I will tell you that that's precisely what was done here. These two gentlemen used Shargav's rules. They borrowed some data, and they generated a model that resisted falsification, and that's why it was accepted very rapidly. Every experiment that was done was consistent with the story. As a counter this example, Linus Pauling, giant of the 20th century, ended up with a 300 helix because he did not have access to the crystallographer. That structure was rapidly invalidated. I got to tell you a little bit about light scattering. Light scattering is the reason that the sun is red, because blue light is removed. The sky is blue because blue light is preferentially scattered, and that is because the scattering function is to the fourth power of the wavelength, and blue light is the smallest part of the visible spectrum. Next time you see a lunar eclipse, you will see that the moon is reddish. The reason is because the light that falls on it has been filtered to remove blue light. If you could do light scattering measurements, static and dynamic, and this will give you a bunch of parameters that you could use to get insight into the capsule. And without getting into a lot of the physical chemistry, I will tell you some of the things that we have learned. So one of the things that we learned is that a single molecule spans the capsule. And we did this by making a measurement in our world, which is a microscope measurement, and a measurement in the quantum world, which is from dynamic light scattering. And we got a beautiful straight line, suggesting that the model involved that a single molecule will grow as the capsule expanded. Now, being good physical chemists, the first thing we do is to try to invalidate the model. And one of the things that we do is ask, is it linear or branch? This is a very tough problem. But you can do it if you have access to light scattering instruments, and you can measure the radius of gyration and the hydrodynamic radius. When things become branch, you have a reversal of the radius of gyration and the hydrodynamic radius, such that the hydrodynamic radius is on the outside. And you could imagine that in a ballerina. A ballerina's radius of gyration will be akin to the center of mass. By the way, I was warned never to do this. That is, never to bring examples from the real world, from the quantum world, because that wasn't right. But I think that I can take some liberty here and give you a sense of what I'm thinking about, whereas the hydrodynamic radius will be the radius of mass and the air around it. And what we find is that the cryptococcal capsule is incredibly unusual and branch. It appears to be a structure that is not a linear polysaccharide, not a simple branch, appears to be a dendrimer. And you can imagine that that's what happens. You have a very large radius of hydration and a small radius of mass. This is the only microbial capsule that is composed of dendrimers, and it raises huge basic science questions on how do you assemble these structures and get them out. And in fact, we have now modified this view to think that this is a dendrimer and that the capsule grows through these large fibrils that eventually create an exclusion zone for India ink. Why care? Because branching affects phagocytosis, antibody binding, oxidant quenching, and interactions with the cells. So it's very, very biologically relevant. Now, in collaboration with Peter Williamson, you can see that we have now even generated a new modification of the capsule. He had the unusual result that he knocked out an enzyme that he did not expect will be infecting the capsule, and ended up with a very large capsule. And these mutants are very different than the original one. And I guess what I'm getting at here is that the power of combining different things, combining genetics with biochemistry, physical chemistry, to get new insights. Now, visions of the capsule. Is it cotton candy, styrofoam ball, or a soccer ball? How would you get at something like this? Actually, if you begin to think about it, that is itself a very difficult problem. So working with some biophysicists, we took a novel approach to do this. You take Cryptococcus and you immobilize it, and then you put a latex bead on it, and this is what happens. You can make all kinds of measurements, and you can see that you can move the crypto away from it, and at some point this will snap. And from that you can calculate the Young's modulus, and that gives you a sense of what this capsule is like. And from that we can say that when they are young, they appear to be a little bit like the capsule is very fluffy. As it gets old, it becomes firmer, and if you ever put antibody on it, it becomes like a soccer ball. It becomes very rigid, and in fact the protective antibodies are the ones that mediate this change. The non-protective antibodies do not cause that change. So now I'm going to switch over to the world of melanins. So melanins, most important is melanin is a pigment. It is not a protein. It is something composed of cross-linked phenol and indole groups. It is found in all biological kingdoms. The only ones who lack it are viruses, although I wouldn't be surprised if we find one of these giant viruses that makes melanin. The ink of the squid, pigmentation, the color of your skin is determined by the melanin granules, and certainly fungi make melanin. Here's a banana trying to repair itself with something that contains melanin. Cryptococcus makes the same melanin that is found in your skin. This melanin is deposited in the cell wall and can protect it against many types of insults, from antifungals, and we have shown that this is a cause of acquired antifungal resistance to heavy metals, to defense things, to pretty much everything. It's non-specific armor. But what I'm going to tell you about is a neater story, and it came from what I think we should all be reading outside our fields. So I was reading about Chernobyl, and it turns out that in Chernobyl there was a chemical explosion in 1986 that caused this structure to become radioactive. So the old Soviet Union could not shut down the reactors. They had 3 other reactors at the site, so they created what they call a sarcophagus around it. But this structure is in danger of falling apart, and I'll get back into it. So They are now they are now going to create something to be in the outside, so when this crashes down and all the dust gets out, that at least be contained with it. So they send in a robot in there, and it collected samples from the spent cores and found there were 37 fungal species growing in the reactor, of which all of them were highly melanotic. Now, that suggested that perhaps melanin was protecting against radiation, and it does protect against radiation. It's remarkable. But this couldn't be the explanation, because even though you're lethally radiated, if you walk into the area, these fungi can tolerate that radiation. The most interesting result was that highly radioactive areas were turning black. The soils were turning black, and they would do so because we thought that the microflora was being replaced by melanotic organisms. So without going through a lot of work, I'm just going to tell you that after 5 years, we thought we established that melanin was allowing the fungi to convert electromagnetic radiation to food, so that the fungi were using radiation for food. And when you think about it, it shouldn't surprise you too much, because biology can always use an electron. And we show, for example, that if you take melanin and you irradiate it, that it will allow the oxidation reduction of NAD and NADH. So we thought we had a major discovery. And we sent it to a single-word journal. And the single-word journal reviewed it, and after 6 months gave us a lot of work, and then after another 6 months rejected it. And then we decided, gee, there are other single-word journals out there. So we went to another single-word journal, and the other single-word journal did the same thing to us. But one of the there were many memorable reviews, but one of the most memorable review was a reviewer who wrote something to the extent of, I can find nothing wrong with the work, but I don't believe it. What do you do? I mean, anyway, I ask you the question, if you had a choice between being in the science parts, in the report parts of a single-word journal, or being in the news, what would you take? Think about it. It's actually an important question, which I'm about to show you, because even though we ended up publishing in PLOS ONE, they had to all of it cover it. So we didn't make it into their science parts, but we made it into their news part. But it may have been good, because recently we found this very interesting relationship, Ferric Fan and I, between the retraction index and the impact factor. And the single-word journals tend to be in this area. Science went on and reprinted it and challenged us. Reprinted it saying that perhaps it was because more people read the journals. And we were kind of skeptical about that, so we decided to follow up the study. We, of course, thought that everyday science is often a messy day affair. They may encourage also to manipulate their data to meet this expectation. And recently we have found, unfortunately, that misconduct accounts for the majority of retracted publications. And we have found that males are responsible for a lot of this. Now, for the females, there is a caveat in the study. All that we show is that the females are missing. It doesn't mean that they don't commit misconduct at the same rate and they just don't get caught. So it's in the discussion. It's out there, something to think about. However, I really wasn't bothered too much. Some of the reviewers wanted you to give them the entire mechanism. Another reviewer wanted you to track an electron down all the way to ATP. And I say you can't do that for chlorophyll today. But one person that gave me a criticism that bothered me told me that, Arturo, X-rays go through everything. How is gamma rays going to be interacting with melanin? And the solution to that came from this individual, Charles Turek, who we collaborated as an electrochemist, and he made a melanin electrode. And then when you turn on the radiation, he got a current. There is no way out. Melanin can interact. If you can get a current, it can do so. And we published it in another one-word journal, but it's not one of the ones that you usually submit to. I will tell you that this has now been confirmed by another group, and we're very happy about this independently. And we have extended it into the visible range. So we think that fungi have a limited capacity for primitive photosynthesis, and that they can, in fact, maybe that's why they express all these pigments. And here is something I took when I was traveling. These are lichens, and you can see the melanized next to the ones that have chlorophyll. We think that these are capturing energy. And here's another picture from Antarctica. In Antarctica, some of the hills, the mountains are completely black, and they are completely black with melanotic organisms. And I ask you, what do you think is going on there? And I think it's a form of photosynthesis. Here is another fascinating thing that melanin can do. If you put melanin, black mushrooms, or into a mouse, you can lethally radiate the mice and they can survive. And if you take the melanin and you mix it with the white mushrooms, you get the same result. What happens is that melanin protects the gut. And by protecting the gut, allows the mice to have probably an auto transplantation. So it protects the gut, they don't get septic, and then they get reseeded from, and we are developing this. We hope to go into clinical trials. It has tremendous potential usefulness in radiation therapy and radiation protection. And last year we sent all this data to the people in the damaged reactor in Japan. But here is something relatively simple that could have tremendous use. I will go back to the fungal infections and the big picture. So the fungi are found primarily, most of the fungal diseases are found primarily in the equator. And that's consistent with fungi that would have evolved to be more thermal tolerant. Now, all the studies predict that the climate is going to get warmer. As the climate gets warmer, the difference between you and ambient temperature is going to become smaller. Fungi can adapt, and we may have many organisms that are today non-pathogenic become pathogenic. Here is an experiment that was done by a company. They basically wanted to develop a fungus to kill insects. And what they found is that the insects are pretty smart. They sun themselves. And as soon as they sun themselves, they fry the fungus. So what they did is they adapted, after only a few months, the fungus to be able to grow at 37 degrees. If this can be done, and you have all the species out there, and the climate is going to go warmer, I think this century the situation may change dramatically with regard to fungi that could take down immunologically intact humans. One of the things that we try to do is we have proposed that in fact virulence is an emergent property that is not predictable. And we set out to do some experiments in this regard by comparing 3 cryptococcal species, but they're very different in thermal tolerance. So we had to go to a host that allowed that. And what we found was that even though all the studies in Cryptococcus will have suggested that these were the critical virulence factors, capsule, melanin, phospholipids, and urease, that in fact was not predictive. In fact, the most virulent organism turned out to be at the lower temperature, the one that did not have a capsule and did not have melanin. The point of this is a very simple experiment, but I don't believe that there is going to be predictability. I believe that into the future there may be probabilistic models, but I don't think that there will come a day when you have an entire genome of one, an entire genome of the other, you know all the epigenetic changes, you know the immune system, and you're going to be able to say this is a pathogenic organism. Last thing I want to leave you with is the question, is virulence a chaotic system? So chaos was pretty much has been discovered a few times. It probably was discovered by Newton, who said that the 3-body problem in gravitation gave him a headache. Poincaré tried to publish a solution to the 3-body problem, and it turned out to be wrong. The bottom line is the 3-body problem cannot be solved because small differences on 3 bodies result in the system becoming unstable in time. The one that this is credited to is Edward Lorenz, who noticed it was an accidental discovery of chaos because what he had was an early computer, and he was doing mathematical models of simulation. And what he was letting the computer run, but he wanted to shorten the experiment. So he began to take the values 3 or 4 days later. What he did not know was that the computer truncated the numbers because it did not have capacity memory. And he could not, and basically, as he will put them together, things will be fine for a while, and then he will have pretty much complete helter-skelter. And this became what is the basis of chaos. Chaos means that small differences in a system will have big differences out there. And if you think about it, this is a fundamental aspect of all biology. Whether you're studying an immune response, whether you're studying virulence, that has never really been looked at before. It's in fact very difficult to look at the problem of chaos because you cannot redo the experiment over and over again. So our system, Galeria, Cryptococcus, and 6 experiments with Galeria, and I'm not going to take you through the math, but I'm going to show you the result. So we were, this has just been published, we were able to find no chaotic signatures in this interaction. But the important thing here is not the result, that there is a method to ask the question. Think about it. We can make vaccines very easily to some organisms, and we cannot make them to others. And we don't really understand that. Wouldn't you want to know whether the system you're working with is chaotic or not? If it was chaotic, you would have a very different view of viewing it. In fact, if the weather was not chaotic, we could have weather prediction far into the future. What happens is that small differences translate into big changes. We have been able to compensate a lot of that by computing power. But something so fundamental has never really been asked. Our immune response is chaotic. Does it vary with inoculum? And I think that this is an area that if we knew the answers to this, it may in fact guide a lot of how we go forward. So I'm almost at the end, and I promise you all we'll come back to the questions. So why are we here? KT event and fungal selection. Why are we so hot? To keep the fungi away. Why do we eat so much? To maintain this very high temperature. Mammals make no sense. Mammals were not going anywhere until the KT event. They have been around for probably over 100 million years. It was an experiment that, in my view, was going nowhere, and yet all of a sudden it becomes dominant. And I think for that to have happened, it had to be selected for. And now that begins to explain why they're so resistant to fungal diseases and why this lifestyle that in reality requires a tremendous amount of food and is very wasteful becomes a dominant. So fungal selection kept down the reptiles, and we never had a second reptilian age. So a final thought, mammals make no sense without the fungi. Most of your caloric uptake is to keep your body temperature. You eat probably 4 or 5 times a day to keep the fungi away. I'll point out to you that emerging diseases in amphibians can be cured by elevated body temperature. You can take the frogs into a 37-degree room, and just putting them in a 37-degree room completely eradicates the fungus. You can go into a cave and take the bats and bring them into the lab and just feed them so that they can get their metabolic rate up and they can clear the fungal infection. So perhaps global warming will save the frogs by roasting the chytrid. And I'll leave you with that image that perhaps somebody will benefit from all that pollution. Thank you very much.
That was amazing. Questions or comments for Arturo?
At least they're not running me out of town.
No.
Ron. Hi, Ron.
Hello.
Last time you asked me an immunology question.
No. I'm going to ask you an editorial question.
Yeah.
So when the reviewers of a paper come back and say your data don't look very reproducible, you explain it as chaotic.
Actually,
is that the result?
No, I'm sorry. Chaos is reproducible. Chaos is both deterministic and reproducible. One of the great problems that we have with chaos theory is that the word chaos in our everyday language doesn't mean the same thing in mathematics, and that introduces a tremendous amount of uncertainty in how we speak.
Now the serious part of the question, which is we've all, especially having just done our ethical conduct of science training today, aware of this issue of reproducibility in science and the publications recently about the irreproducibility of science. But as you said, very small effects can give you very big differences. So do you think a lot of the irreproducibility is not the make-it-up kind that you pointed out here does occur, but the fact that we are not as careful as we might be about whether we really are precisely replicating an experiment, and we're getting into these regimes where very small differences give you very big readout differences, and we should use that as a guide to better understanding the biology and how things diverge?
So Ron, I think you're hitting on something really, really important, and it's something that affects everyone in this room. And you just take a couple of minutes. So we have been looking at the issue of retractions. Retractions are evidence for failure in science. Even though retractions are good that you can retract it and correct science, think about what's going on. So the way we got this data, by the way, is we reviewed every single retraction notice out there. We found that the retraction notices were often fraudulent, in that people lie in the retraction notices. And it was basically, they said, the paper is being retracted because I can't reproduce table 2. And then you correlate the names with the Office of Research Integrity, and you find that a lot of that was misconduct. So one of the things that we're doing to address your question is we're now going back and analyzing the honest retractions, going one by one to try to see patterns. And I think it's a complicated story. One of the most common causes of error is contaminated cell lines. And contaminated samples often lead to multiple retractions. And I think the answer to some of this is yes, some of it can be error. It's also, we are under tremendous pressure of funding. Also, careers are being made whether you publish in a single word journal or not. And you can imagine the pressures that it's putting on people to often not necessarily put the whole data out there. Part of the reproducibility. I wouldn't go as far as calling that misconduct. But I think that we have really messed up priorities in science, including the fact that we judge the value of the work based on where it's published rather than the content. So it's a perfect storm. It's funding problems. It's the fact that we're judging work very differently. It's chaos in the numbers. It's the fact that people often publish their best data. Why don't you publish a worst experiment? Because you can't get it published. And also, if you try to reproduce somebody else's data, nobody wants to publish your work. So I think that what you're getting at is almost, we need to, as a scientist, we need to sit down and we need to basically try to work through these things out, because the literature, which is our lifeblood, is in trouble. And this may be part of it, chaos, but I think there's a lot of other issues out there. Incidentally, did you see the paper that says that, you know how basically we judge significance at 0.05, 0.05? It's been argued that in biology, with all these changes, that we should be a lot more stringent and use a greater significance before we accept the 1 in 20. So it's a lot of things that I think we need to, this is a big discussion, a big issue, and we need to, as a community, begin to address it. Yeah.
You mentioned several fungal diseases that are extinguishing or almost extinguishing cold-blooded and warm-blooded species. The frog disease, which I can't pronounce, and
Chytrid, right?
the white-nosed bat syndrome. Is there any way you can say that some emerging infectious fungal disease won't begin to extinguish human beings? Are we immune to that?
No, we're not. And in fact, one of the things that we have been doing is we're taking that collection and we have been looking at where are the organisms that are thermotolerant. And thermotolerance appears to be spread throughout the fungal kingdoms. Some of these organisms are completely resistant to our antifungal agents. And if one of those emerges, we are going to have a huge problem. But now, I don't believe, because there are plenty of fungi, there are 1.5 million species, and at least 5 to 10% of those can live at temperatures greater than 45 degrees. So the potential number of organisms out there is enormous. And if you don't believe that this can happen, go talk to the frogs. Or to the bats, who were doing well until this thing got introduced into the United States.
That was the most elegant argument for increasing funding for fungal diseases that I've ever heard.
I didn't say anything about that. Yeah. Well, the reason that it was associated with global warming was fascinating, because what happens is that as the planet gets warmer, it climbs up the mountains. So what you see is that that it climbs up, it used to be that it's restricted primarily to the areas around sea level. Some people think that this was spread when xenopus was sent all over the world, because in those days it was used for pregnancy testing. It was an example of that. I think it was pregnancy testing. Somebody could correct me on it. But the idea is that we pretty much spread it already, and humans were the vector for disseminating it, right?
So, as immunologists, we do a lot of stuff in mice.
Yes.
And most of our mice are housed at below the temperatures that they want to be housed at.
The Chris Karp's paper in JEM.
Yeah, so how much effect do you think this is having as a broad principle in all of immunology? If we're off in so many ways, I mean, the implications of it could be really profound, right?
Absolutely. I mean, the argument goes like this. Mice normally lives at temperatures on the ground in which they huddle together of 37, 38, even as 39 degrees. When we adapted it, we adapted it into our temperatures because we don't want to go into a warm room to do a mouse experiment. So now we have mouse, we have 100 years to selectively breed mouse that are cold stress. And it raises the question whether some of the problems in relating some of the immunological studies in human to mice may have the origin in the fact that we are working with a cold stress organism. And I think the jury's out there. I know that mice are getting a lot of bad rap recently, but I think it's worthwhile to think of the many ways in which they have also guided us correctly.
Mammals have been here, I guess, the dominant species for maybe 65 million years?
No, much longer.
Oh, much longer. Okay. So in any case, what have been the constraints on fungi to evolve that thermal tolerance over those millions of years and not be as good as bacterial viruses at infecting mammals?
You know, I think you're thinking of virulence almost from a mission. The fungi don't care. I mean, they live in the ground, they digest stuff. When they go into an animal, they go into a new ecosystem. And it just happens that through selection, they have been fighting with protozoa and bacteria all this year, and they're selected for traits that can occasionally cause disease in mammals. But I don't think that necessarily you should think of it as them evolving to have thermotolerance. We don't understand why some are thermotolerant. We can explain it. For example, the organisms that live in a compost can get pretty hot, and they survive it there. But I don't think that the virulence is an outcome. It is hard to argue that an organism will become thermotolerant so it could go into another site.
Thank you.
No, you're welcome.
Okay.
All right.
Thank you very much for an extraordinarily stimulating lecture.
Thank you.
That was fantastic. It was really like it. It was really absolutely wonderful. Enjoyable.